Uh, Dear colleagues, thank you for the opportunity to present our work on using machine learning to decipher genome regulatory grammar. Uh, Actually, it turns out that while the main focus and main progress for now is done in predicting the effects of coding invariance in genome, the main part of phenotype-associated single nucleotide variants are non-coding. And we do know that even single nucleotide non-coding mutation can result in developing such severe disorder as Parkinson's disease by changing the binding of one transcriptional factor. So, to solve this problem, genome-wide models were introduced. The main idea behind them is relatively simple. Let's learn a model to predict epigenetic signals like chromatin accessibility, histone modifications, pitch experiments, and so on, given a nucleotide sequence. The most recent models using that paradigm are Informer and its successor, Barzoi. So, how to use such kind of model to predict the effect of mutation? Again, the idea is pretty straightforward. First, obtain epigenetic tracks predictions for both reference and mutated nucleotide sequences. Next, one can aggregate these predictions using some simple aggregation methods like maximum of difference between corresponding tracks or the difference between tracks closest to target cell line, and thus you can acquire a score presumably correlated with expression change. Also, the possible solution for that is for creating data is to train additional model like lasso regression or random forest to predict final expression given epigenetic tracks predictions. After that, you can be published in Nature, and there are papers in Nature on that. And some results are pretty amazing. For example, such methodology allows us to predict the results of regulatory sequences iteration mutagenesis. Predictions correlate for regulatory elements uh, quite well, but yet the method completely misses some binding sites. Uh, Actually, these results can be further improved by fine-tuning models on saturation mutagenesis data. But as we shown in our previous work, training on such kind of data is complicated due to the data leakage. So anyway, problem solved, and we can happily use this instrument and forget about anything else. Is it true? Unfortunately, no. It turns out that while models' predictions correlate with ground truth, then measuring across loci of the same individual genome, then it comes to ranking the effect of mutations in the same locus across different genomes. The correlation is sometimes is insignificant, sometimes is really negative, and the distribution of correlation across different loci is uh, concentrated around the zero. Moreover, it becomes increasingly evident that such models are good at predicting only conservative peaks, but fail to predict cell-specific signals. One of possible explanations for that is the genome is not as big as we used to think. Native genomic DNA represents only a small fraction of the space of possible genomic DNA. Renal genome is full of repeats and homological elements, and the number of common chimeras between 200,000 nucleotide long sequences differs from expected by random chance by a huge margin. This results in model inability to distinguish between regulatory sequence and its neighbors. On the picture, red region is the only one responsible for expression, but there is no information in genome from which model can deduce that, so it instead learns that both red and blue regions are required. One of the possible solutions for this problem is using data from massively parallel reporter assays, allowing to measure effect of it on expression of thousands and millions of random or specially designed nucleotide sequences. And actually, this data can be easily constructed in the way it would be easy for a model to learn causal dependencies. So now our aim is to create a modern model to process such kind of data. Uh, the nucleotide sequence scoring problem can be reformulated in terms both of natural language processing and image recognition. In the case of natural language processing, we treat the whole sequence as a sentence in natural language, where each nucleotide is a distinct word. In computer vision, we use one-hot representation to transform a sequence into one-dimensional four-channel image. And as it has been shown, the latter approach usually is simpler and results in no in sometimes in better results. So we used that approach. Also, for computer vision, the actual state of the art in terms of classification and some other tasks is efficient net v2 model. It has amazing performance, relatively small training time, and the trained model is comparably small, and thus applicable to small dataset with reduced risk of overfitting. Efficient net v2 is a fully convolutional neural network with several sophisticated tricks which can be repurposed to decipher regulatory sequences. So let's have a look at the overall LegNet architecture. The input is passed to the stem block. Actually, it's just a usual convolutional block, followed by the sequence of 6 efficient net v2-like blocks, followed by a mapping block to transform resulting features into prediction. The key idea of a special squeeze and excitation block is to allow the feature channels to communicate with each other and, as a result, adjust their activations. This can be considered as some form of a highly regularized attention mechanism. Also, from multiple studies, we know it is not only the architecture that matters, but also the training scheme. We tried several modern approaches and found one-cycle learning rate schedule coupled with Adam W optimizer to be the most promising during the preliminary studies. We selected the maximum learning rate for this approach using learning rate range test described in the original paper by Smith and Token. As a result, we observed the so-called superconvergence. This approach halved the number of epochs required to train the neural network compared to the other methods, and further increased the model performance. Next, we applied our model to the task of predicting the effect on expression of random 80 nucleotide long nucleotide sequences measured in yeasts. And the results looked very promising. Not only, sorry, and not only our solution achieved top 1 performance on public leaderboard and in the final evaluation of Dream 2022 challenge, but it did it in all subcategories, especially in predicting the effects of single nucleotide substitutions, which is the most real-world used scenario for now. So next, we decided to apply our model to the task of predicting the effect on expression of human regulatory regions, and the results were amazing. Not only we surpassed our solutions trained from scratch, but actually our model was able to outperform fine-tuned genome-wide models in 2 out of 3 cases. And in the third cases, actually, the data set used is quite noisy, so the difference might be attributed to the noise in the data. Also, what is really important, our model was able to predict the results of saturation mutagenesis for human promoters and enhancers, and captured all important regions in them. Next, our collaborators have shown that on Drosophila MPRA data, our model and its modifications yet again surpasses the quality of models from original work. Again, if we apply our architecture or its modification to the task of predicting epigenetic tracks, it turns out that using times fewer number of parameters, our model is able to surpass or perform on par with current state of the art for single epigenetic track prediction. Even if applied to the task of predicting allele-specific events, our model again surpasses the quality of other models used for this task. But this is not the end of the story. If we can predict expression, why not to try to adapt our model to the task of generating sequences with desired expression? We can then use such model To generate cell-specific elements with biotechnology needs, applied for active learning to select sequences whose activity should be experimentally measured to improve model performance. And finally, we can use such a model to perform in silico studies, allowing to decipher genome regulatory grammar. And it turns out that we can modify recently published cold diffusion process to train our model to restore mutated sequence given its effect on expression. After that, we can use model trained in this way to generate sequences with desired expression. Uh, Firstly, we validated our approach in silico setting, and the correlation between requested expression and expression predicted by predictive model is quite strong. But we have upcoming in vitro validated results. Unfortunately, I can't share them for now. That our diffusion allows not only to generate sequences with desired expression, but actually is able to generate sequences with cell-specific activity. Thank you for your attention. I will be glad to answer your question.